Hello, this is Jay Kolls from Tulane University, and on behalf of my co-chair, Dr. Geraldine Butler from the University of College Dublin, we welcome you to the October 28th MycoTalks. We have two excellent speakers today. Dr. Betty Wu-Hsieh from National Taiwan University is going to talk on differential usage of receptors for fungus-induced innate cell responses. And then Dr. Salome Leibundgut-Landmann from the University of Zurich is going to talk about immunosurveillance of fungal commensalism. Here's the organizing committee, and the philosophy of MycoTalks is to foster diversity in terms of career stage, gender, geography, and field of study. We have two 30-minute talks on all aspects of medical mycology for the research community, followed by 30 minutes of in-depth discussion. Please submit questions through the Q&A function, which should be down at the bottom of your Zoom screen. And also mention which speaker you're directing your question to. And then recordings will be available on the MRC's Center for Medical Mycology YouTube channel. For upcoming MycoTalks, remember you have to register for each session. So you should be getting an email link to register. But in November 18th, we have Dr. David Denning from the University of Manchester, and Dr. Anita Seal from the University of California, San Francisco. And then in December, we have Dr. Axel Bargaj from Hans Noll Institute in Germany, and Dr. Ashraf Ibrahim from the Linguist Institute in the US. And then we have a couple series for, we have one series for trainees. This is the Medical Mycology Trainee Seminar Series. It's for all students, postdocs, and clinical trainees, as well as junior faculty. It gives you an opportunity to present your work virtually to this global community. These talks are on the second Thursday of the month, but timing can be adjusted to fit speakers in their time zone. It helps us highlight the scientifically broad and international medical mycology community by nominating speakers with wide scientific interest and global diversity. So you can get in touch and submit your abstract here at this link highlighted here on this slide. And maybe Jules can also put this in the chat for those that are interested for this series. And then we have the medical mycology clinics, and we have a really exciting series of talks for the Friday, December 3rd, and this is on COVID-19 associated pulmonary infections, where we have Joost Walters talking about the COVID-19 associated pulmonary aspergillosis, and then Rita Aurora talking about the management of COVID-19 associated sino-orbital mucormycosis, which has obviously been a very topical issue since the COVID pandemic. So with that introduction, I will turn it over to my co-chair, Dr. Geraldine Butler, who will introduce our first speaker.
Hi, so it's a great pleasure to welcome Professor Betty Wu-Hsieh, who is joining us very late at night from the National Taiwan University College of Medicine. So Betty received her PhD from the University of California in Los Angeles, then moved to Columbia and back to UCLA before finally moving to set up her lab, the Institute of Immunology in Taiwan. So she's recently retired as a full professor and is now serving as an adjunct professor, so not ready to stop working yet. And since graduate school, Betty has worked on histoplasma capsulatum, particularly the interaction with the immune system, and more recently studying the interaction of the immune system with Candida albicans. As well as fungal immunology, she's also worked on dengue virus and SARS-CoV. So it's a great pleasure to listen to her present today. Thanks, Betty. You're mute, Betty, could you turn on your microphone?
Thank you, Geraldine, and thank you for inviting me to MycoTalks. It's my great pleasure and honor to speak to a large audience across the globe over the internet. What I'm presenting to you today is a summary of work from my laboratory over a number of years. I will present to you the key findings of each separate study and discuss the insight gained concerning innate cell receptor usage. I will borrow two illustrations from Neil Gold's review article. First, to show you fungal cell wall. Fungal cell wall is composed of distinct layers of carbohydrates, having beta-glucan and chitin as the basic inner cell wall structure. What's exposed in the outer cell wall layer is what is being recognized by innate cells to effect cellular function. And fungi come in different shapes and sizes, which directly affect their interaction with innate cells. The innate cells I'm going to tell you about from our study is macrophages, dendritic cells, and neutrophils. The two organisms that I am going to describe for you in innate cell usage is Histoplasma capsulatum and Candida albicans. There are two chemotypes in histoplasma. Chemotype 1 expresses beta-glucan in the outer cell wall, whereas alpha-glucan is masked. Whether the organism is live or heat kill, they express beta-glucan. Whereas chemotype 2, their beta-glucan is masked. Alpha-glucan is expressed. Only when the gene for alpha-glucan is knocked out, beta-glucan is expressed. The Candida albicans, when it's heat kill, they expose beta-glucan, and live Candida albicans, beta-glucan is masked. And my study employed Histoplasma strain 505, which belongs to chemotype 1, and its beta-glucan is expressed, and also use Candida albicans strain SC5314 for our study. This diagram illustrates what I'm going to tell you. To study receptors utilized by histoplasma on macrophage and dendritic cell for functions like phagocytosis, TNF, IL-6 production, and IL-1 beta production. For Candida albicans, I studied albicans' interaction with neutrophils and what receptors it utilized to trigger netosis. Macrophage plays multiple roles in its interaction with histoplasma yeast. It serves as a host cell where it takes up histoplasma, allowing it to replicate. By replication, histoplasma induces macrophage apoptosis. Apoptotic macrophages carry fungal antigen, deliver it to dendritic cells, and dendritic cells can cross-present fungal antigen to activate CD4 and CD8 T cells. Macrophage also serve as effector cells. When activated by interferon gamma and TNF alpha, it can restrict the intercellular growth of the organism. Both dendritic cells and macrophage can function as cytokine-producing cells. Let's look at receptor expression profiles in different macrophages. Thioglycolate-elicited macrophages express high levels of CR3 and dectin-1, and almost 100% of the cells express these two cytokines, whereas other populations of macrophages express lower levels of dectin-1 and with less percentage of cells. Thioglycolate-elicited macrophages is what we use for our study. We first want to know which receptors mediate phagocytosis of histoplasma. We physically label histoplasma and use flow cytometry to analyze what percentage of cells take up histoplasma. And as you can see, is depending on the histoplasma to cell ratio. It can take up to about 80% of the cells can take up histoplasma at high histoplasma and cell ratio. To determine the receptor that's responsible for phagocytosis, we use reagents like laminorin, menin, and antibodies, Dectin-1, TLR2, TLR4, and the rest of the receptors. None of these reagents block phagocytosis. With antibody to CR3, 5C6, there is a blocking effect. At 1 microgram per ml of antibody, it blocked about 40 to 70% of phagocytosis. And increasing concentration of the antibody did not further reduce phagocytosis. By using receptor deficient macrophages, you can see that CR3 deficiency reduced phagocytosis, whereas Dectin-1 deficiency had no effect. Double deficiency is not much better than CR3 deficiency. Therefore, CR3 is a receptor that's responsible for macrophage phagocytosis of histoplasm. The story with cytokine response is different. Single receptor deficiency reduced cytokine production by about half. Double deficiency further reduced TNF and IL-6 production. Therefore, there seemed to be an additive effect of CR3 and Dectin-1 in TNF IL-6 production. To study the relationship between CR3 and Dectin-1, we look at these two receptor relationship after the cell engulfed histoplasma. As you can see, 15 minutes after encountering histoplasma, there's a co-localization of Dectin-1 and CR3 together with the lipid raft marker, CTP. By sucrose gradient fractionation, we can also see that CR3 and Dectin-1 appear in the fractions where lipid rafts are. Therefore, these two receptors are associated in lipid rafts when they encounter histoplasma. And lipid raft is important for TNF production because removing cholesterol reduced its production, replenish the cholesterol, restore its response. And by micro-Western analysis, we identify the signal molecules that are activated after macrophage are stimulated with histoplasma. These are the ones that are activated. And without going into details, I'm just going to tell you that SEEK, ERK, JNK, and AP1 activation are responsible for macrophage cytokine production. The differential, receptor usage of CR3 and Dectin-1 are reflected in the animal infected with histoplasma. Single deficiency of either receptor reduce its survival, and double deficiency further reduce its survival, and also reflected in cytokine production, TNF, interferon gamma, IL-17, and not much for IL-6, that single deficiency reduce cytokine production by half, and double deficiency reduce it even further. So from this study, we learned that CR3 is responsible for phagocytosis of histoplasma. Dectin-1 is not involved in phagocytosis, but participates in cytokine production. Both CR3 and Dectin-1 participate in TNF-alpha IL-6 production as equal partners. The next question is, how is Dectin-1 different from CR3 in TNF and IL-6 production? Before going into that, I'll give you a brief introduction of LC3 associated phagocytosis, LAP in short. LAP is initiated following recognition of pathogen by pattern recognition receptor. The pathogen resides in a single membrane phagosome. The phagosomal membrane is enriched with LC32, which is a lipidate form of LC3. Western blot analysis showed that histoplasma stimulation induced LC32 conversion to the lipidated form. And the vesicle that contains histoplasma is single membrane. In unstimulated macrophages, LC3 is diffusely distributed. After encountering histoplasma, LC3 is increased and localized to the phagosomal membrane. Therefore, there is a lipomosome formation after macrophage take up histoplasma. Dectin-1 deficiency decreased histoplasma-induced LC3 formation, and neither Dectin-2 deficiency nor CR3 deficiency affect LC32 conversion. And this is also can be seen visually that Dectin-1 deficiency, there's no LC3 recruitment nor increase in cells after taking up histoplasma. Laminorin can dose-dependently reduce histoplasma-induced LC32 formation. Using DPI, which inactivates NADPH oxidase and NCF deficient macrophages, we found that inactivating NADPH oxidase reduces or inhibits LC32 conversion. And this means that NADPH oxidase is essential to histoplasma induced LC32 conversion. Okay. LC3, silencing LC3 does not affect Seq phosphorylation, but reduces signaling molecule junk, ERK, P38, C-Jun, and C-Phos phosphorylation. Moreover, silencing LC3 affected or reduces TNF IL-6 and IL-1 beta production. So from this part of study, we learned that Dectin-1, but not CR3, triggered TNF IL-6 production, involves NADPH oxidase dependent LC32 conversion. Therefore, CR3 induced cytokine production and Dectin-1 induced cytokine production, although the outcome is the same, but they go through a different pathway. The next question is, how does dendritic cell uses receptor for IL-1 beta production in response to histoplasma? Let's look at dendritic cell receptor expression. You can see that bone marrow derived dendritic cell express good levels of Dectin-2. 76% of cells express Dectin-2, which is not seen in other macrophages. And Dectin-2 expression is relatively low compared to macrophages, but there is about 52% of cells express Dectin-1. Stimulation of dendritic cells with histoplasma induce IL-1 beta production as early as 6 hours and increase at 12 hours and on. This IL-1 beta production is caspase-1 dependent because using a caspase-1 inhibitor dose-dependently reduces IL-1 beta production. And IL-1 beta production is NLRP3 dependent. NLRP3 deficiency completely abrogate IL-1 beta production, meaning that IL-1 beta going through inflammasome activation is produced after NLRP3 associated inflammasome activation. And Dectin-2 deficiency reduces histoplasma-induced IL-1 beta production, whereas Dectin-1 deficiency had no effect at all. However, double deficiency further reduce IL-1 beta production. So what is the role of Dectin-1 here? We use antibody blocking, reciprocal antibody blocking assay to look at this phenomena again. As you can see here, as expected, Dectin-1 deficiency did not affect IL-1 beta production. However, blocking Dectin-2 in wild type cells, as expected, reduced IL-1 beta production, and blocking Dectin-2 in Dectin-1 deficient cells further reduce IL-1 beta production. The same here with Dectin-2 deficient cells. In Dectin-2 deficient cells, blocking Dectin-1 also further reduce IL-1 beta production. And this relationship also shown in signaling pathway. It affected Dectin-2 deficiency reduces seq and junk phosphorylation, whereas Dectin-1 deficiency had no effect, and double receptor deficiency further reduced these signaling molecule activation. So from this study, we learned that Dectin-2 functions as a primary receptor for dendritic cell IL-1 beta production. Dectin-2 serves as a secondary receptor, taking the role to respond in the absence of Dectin-2. And both Dectin-2 and Dectin-1 trigger signal 1 for pro-IL-1 beta production and signal 2 for inflammasome activation, which I didn't have time to go into the detail. Okay. And the last part of my talk concerns Candida albicans interaction with neutrophils. Netosis happens when stimuli or pathogens stimulate neutrophils. Neutrophils undergo a process that the nuclear envelope is disintegrated, chromatin condensation, plasma membrane rupture, and release of NATs. This can be assayed by the presence of extracellular double-stranded DNA in the supernatant. Taking up Candida albicans, whether it's opsonized by serum or unopsonized, there is a membrane, nuclear membrane disintegration and cytoplasmic membrane disruption by 1 to 2 hours after encountering the organism. compare opsonized Candida and unopsonized Candida induced netosis, we found that opsonized Candida induced a higher percentage of netotic cells than unopsonized Candida. And it is the hyphae form that induced double-stranded RNA release in both situations. The requirement for NADPH oxidase produced ROS is different between opsonized and Candida, unopsonized Candida induced netosis. Opsonized Candida induced netosis required NADPH oxidase. In deficient cells, there's a reduction of double-stranded DNA release. And this is also for unopsonized Candida albicans, NADPH oxidase is not required to induce netosis. The receptors that's involved in both situations are different. Unopsonized Candida requires dectin-2 for induction of netosis, whereas opsonized Candida requires CR3 for induction of netosis. We used a mouse model to study netosis in vivo. Mice were injected with 2 doses of casein, 18 hours apart. 4 hours later, we found 78% of peritoneal cells are Ly6G positive neutrophils. Using this peritonitis situation, we injected mice intraperitoneally with Candida albicans and Cytox orange. Cytox orange is a cell-impermeable DNA dye. By tracking the appearance of Cytox orange, we can trace the formation of nets in the peritoneal cavity. We also use D-tomato expressing Candida albicans to track the presence of Candida in the peritoneal cavity. By 1 hour, there's large quantity and 2 hours to be detected. Interestingly, by 2.5 hours, there's a reduction of Candida albicans in the peritoneal cavity. In the meantime, there is an increase of Candida in the kidney, which means the Candida is spread from peritoneal cavity to the kidney. If we treated the animals with micrococcal nuclease, which is a non-specific DNA degradation enzyme, treatment with micrococcal nuclease abolished the effect compared to heat kill inactive enzyme. We discover that there's less Candida in the peritoneal cavity, but more in the kidney, which means netosis, if you inhibit netosis, then the organisms spread to kidney, more organisms spread to the kidney. Dectin-2 deficiency also affected netosis. We use Ki67, which is a marker, cell cycle marker for netosis, using that as a marker to track netosis in animals. In Dectin-2 deficient mice, there's less net formation in the peritoneal cavity, as well as in the mesenteric tissues. Dectin-2 deficiency also reduced Ki67 positive neutrophils in the peritoneal cavity. And there is less Candida at 3 hours after infection, less in peritoneal cavity, and more is spread to the kidney. And this phenomena is netosis dependent. So let's go back to this diagram. What we discover here is that histoplasma interacts with macrophages through CR3 to be phagocytosed. And through dectin-1 and CR3, it produces TNF and IL-6. Interaction with dendritic cells through dectin-2 and dectin-1. Histoplasma induce IL-1 beta. Candida albicans interacts with neutrophils through dectin-2 if it's unopsonized, through CR3 if it's opsonized to induce netosis. So what we learn from these studies are following. Each innate cell type exhibits unique receptor distribution profile. Engagement of one receptor may trigger more than one cellular function, just like CR3 can function as phagocytosis and cytokine production. Two receptors may collaborate as equal partners for the same cellular function, like CR3 and dectin-1. They collaborate to produce TNF and IL-1. And there is an order of priority exists for differential receptor usage. Like dectin-2 functions as primary and dectin-1 as secondary receptor to mediate IL-1 beta production by dendritic cells. Two different receptors recognizing different ligands may result in a similar outcome, like engagement of CR3 by opsonized Candida and dectin-2 by unopsonized Candida. They separately result in netosis. Thank you. And this is the people who in my lab contributed to this study. Zhixuan Lin, Junhua Huang, Shenyang Wu, Zixuan Zhang. And also I'd like to thank Jenny Ting, Gordon Brown, Mihai Ni, Netia for their generous gift of knockout mice, and my colleagues in National Taiwan University and National Health Research Institute for their technical contributions. Thank you.
Thank you, Betty. That was a great talk.
Thank you.
All right, so if you have questions for Betty, please enter them in the Q&A session. We already have 3, and we'll get to those in the discussion portion. And so let me move on and introduce our next speaker, Dr. Salome Leibundgut-Landmann. Salome has been a leader in studying fungal commensalism, and particularly the T cell response to fungi ranging from Malassezia to Candida. Salome got her PhD in the lab of Professor Walter Wythe at the University of Geneva, followed by a postdoc in Professor Caetano Risa Souza's laboratory, and then another postdoc in Annette Oxenius at the Institute of Microbiology in Zurich. And she's now faculty, associate professor at the Vet Suite faculty in Switzerland. And as I mentioned, she studies T cells in response to Candida and Malassezia, and she's made some seminal insights into the role of a recently novel population of T cells called tissue-resident memory cells. So she's going to talk about commensalism and immunity to fungi. Thank you, Salome.
Thank you very much, Jay. I assume my slides can be seen. And actually, I just need to redo this because they're not showing properly.
It looked pretty good, but let's try again.
Now it's, I believe it's Okay. Okay. Thank you very much, Jay, for this kind of introduction, and thanks to all the organizers of this amazing series for giving me the chance also to be part of this as a speaker. As Jay mentioned, I'll talk about commensalism of fungi, and especially focusing on Candida albicans in the context of the oral cavity and on Malassezia in the skin towards the later part of my talk, if I manage with time. Now, yes, there you go. This scheme, probably the vast majority of all of you have seen plenty of time. Let me just use it to get started and remind you that all our body surfaces, including the external ones like the skin, as well as the mucosal surfaces, the internal surfaces, are all heavily colonized with a broad variety of different fungi, including on the skin, very prominently Malassezia, various species of Malassezia, and then the mucosal surfaces being much more colonized with much more diverse populations, and whereby Candida colonizes the vaginal tract, the gastrointestinal tract, as well as the oral cavity. And all these various tissues differ greatly in many parameters, including pH, oxygen pressure, microbiota composition, the nature of their epithelial lining, and so on. And these variations, of course, affect very much the nature and the parameters that determine how Candida can, or any of these fungal commensalisms can actually live as commensal there, how they're stably maintained. And in if this homeostasis, if this equilibrium is somehow disrupted, we're probably all well aware that then this results or can result in pathology, which again differs largely on, of course, depending on the fungus, but also very much depending on the organ. Just giving you some examples, with Candida causing oral thrush in the oral cavity, causing a very different form of disease in the vulvovaginal tract, and in the gastrointestinal tract, being prone to translocate and disseminate systemically. Now, it's key to really maintain the homeostasis, and this is true to throughout life, basically, that we really continuously keep this equilibrium, as dysbiosis has pathological consequences at any stage of our life. Now, why is that actually so essential to keep these commensals on us if they're bearing such a threat? Well, there was a lot of speculation for a long time, but more recently, we've actually learned, and there are now nice examples of benefits and beneficial effects of commensal fungi for hosts and the health of the host, with just a few studies that I want to mention here as examples. Examples on the relevance of Candida albicans gut colonization. From murine studies, we've learned that they reduce susceptibility to other pathogens such as Clostridium difficile, but also protect against bloodborne Candida or Staph aureus by various groups that have contributed to this, and also actually propose mechanisms how this happens through mainly the immune response against these against Candida in the gastrointestinal tract. There are also examples with somewhat less mechanistic details on Malassezia and the relevance or the benefits of Malassezia on our skin, such as a recent very nice study from Julie Segre on how Malassezia commensalism actually regulates and contributes to regulating Candida auris skin colonization, or how a study by Tom Dawson, how Malassezia impairs bacterial such as Staph aureus biofilm formation, with a link to atopic dermatitis. Now, is thus become clear that maintaining homeostasis is key, and it's not just important to prevent overgrowth of the fungus on the epithelium, on the barrier, and preventing damage to prevent disease, but it's actually equally important to also prevent elimination of the fungus. Now, let me just make a few considerations about the requirements of commensalism in a sort of a theoretical manner before then going into actual data. Well, for commensalism to establish stably on the epithelium, there are fungal and host factors, and among the fungal factors, first of all, the fungus ought to be retained on the epithelia through adhesive and other processes, and the fungi need to sort of like the environment and be metabolically adapted to this host environment where it lives to actually grow and thrive, but it shouldn't like the environment too much in order, of course, to avoid overgrowth and also virulence factors should be limited, or the expression of such factors should be limited to avoid tissue damage and invasion. Now, all these processes of overgrowth and invasion are very heavily controlled by the immune system, and there is actually a homeostatic, constant activation of the immune response to really control this. And these host mechanisms can be divided in several ones, including the epithelium, which provides, first of all, a physical barrier, but not only, it also provides antimicrobial peptides and other antimicrobial factors. Then there are very important tissue resonance cells that survey the barriers. And last but not least, a key, of course, factors of the adaptive immune system, including first and foremost T cells, and especially those producing IL-17, but also, as we've recently learned from exciting work by Kolls and others, that antibodies, especially IgA, are also essential to really maintain homeostasis on the barriers. Now, given that there is this constant immune activation during commensalism, there is an important aspect also that the fungi ought to be resistant to these host defenses in order to not be cleared. Now, I guess it's very well known in this community that any or several different breakages in these homeostatic immune mechanisms can lead to infections, depending different immune mechanisms breaking, causing different forms of disease. But also what I would like to highlight is that dysregulation of these homeostatic antifungal responses especially in predisposed conditions can also lead to pathology and then to actually immunopathology. And these can be local or in distant organs. Now, I have divided my talk in 3 parts, and I'd like to first of all introduce to you a model of Candida commensalism in the oral cavity that we've established and studied, and which is also the basis then for the second part, where I'll talk really about the T cells that Jay also mentioned in the introduction, that are really, I think, responsible for maintaining as an important host factor the stable commensalism in the oral mucosa. And then finally, if time remains, talk about some immunopathological consequences of antifungal immunity raised against commensals. So we're using animal models, especially mice, in my laboratory to study the fungal host interactions. We use the OPC model, which was first introduced by Scott Filler, but also used by many others in the community, where we sublingually deposit the fungus without any injection, without any tissue abrasion, so no damage that we introduced experimentally. It's important to mention that our experimental mice are actually initially Candida naive, so anything that we do to them, any Candida that we give them is causes a first encounter and the primary response to them. Then the mice are completely normal, not immunosuppressed, not antibiotically treated. And the big advantage of using mice is that we do not have inter-individual differences between individual animals. So we can also study the differences that occur in response to variations on the fungal side in a kind of stably kept host. So when we do disinfection, as many of you have seen in many talks given by various people using that model, if we use strain SC5314, we get a very good colonization of the oral mucosa. But this infection is very rapidly cleared, and this led also to the notion that C57 black 6 mice are naturally resistant, or even mice in general are naturally resistant to Candida albicans. However, we played around with using different isolates of Candida albicans and actually found that mice are not necessarily naturally resistant to Candida albicans, but actually it just varies, depends on the strain. With commensal strains, such as this strain called 101, it colonizes initially the oral mucosa to very comparable levels, but then it actually doesn't get cleared. It persists for days, for weeks, for months. And what is very apparent when we look at what could be the difference for this different outcome, or the cause for this different outcome, that's the initial host response that is raised to this virulent strain SC5314, which is a very imminent and highly inflammatory response, which is also responsible then for this rapid clearance. While in case of this commensal strain, there is barely anything happening that we would be able to detect, and that then allows persistence. Now we were wondering to understand this in a little more details, and we did a transcript profiling of the 2 strains in the oral tissue by using an amplification technique developed by Dominique Songlar, and actually found we also included some in vitro samples. They're not mattering so much here, but what is really apparent is that there is a very clear segregation of the transcriptome of the Harley virulent strain from the commensal isolate. And actually, as this is commensal and persisting in the animal, we could also look over time at different time points, and the response actually further segregates away from the Harley virulent strain. And there were many genes differentially regulated, but the most stunning ones and apparent ones were those linked to virulence and filamentation. And also further functionally confirmed these differences by many phenotypes, including a difference in filamentation. So strain 101 is not that it's non-filamentous, but it's just forming slightly shorter filaments and also less stable filaments, as shown here in an example. And this also then is associated with a reduced capacity to invade tissues, invade epithelial cells, but also really invade the 3-dimensional epithelial tissues. And finally, there is a much reduced expression of many virulence genes, including damage-inducing, the gene encoding the damage-inducing factor Candida lysine AC1. This results in an almost complete lack of induction of damage, and also they're expressing or inducing much less host response genes. Now, in a first brief summary, while this high virulence strain S2E3 is strongly filamenting, inducing a high degree of epithelial damage, this triggers in release of alarmins and really the recruitment of neutrophils, causing a very strong inflammatory response, which in turn results in a very rapid clearance of the fungus from the host. While in contrast, this commensal strain 101 actually evades this immune response by not even inducing it, due to a limited degree of filamentation and no or even on the very low degree of epithelial damage. There are also additional changes between the 2 strains. The commensal strain is very much metabolically adapted to the epithelium, and in particular the keratinized epithelium of the tongue that we analyze. There's increased resistance to host responses and oxidative stress, and this really allows long-term survival in the oral niche. And we've actually looked at many more strains, commensal strains that do not trigger damage, and we find very comparable patterns in those when tested in the oral mucosa. Now we were wondering, can we make strain 101 a little bit more pathogenic, or the opposite, also strain S2E3 less pathogenic, which certainly has been done before, but we focus on 101. What is really, can we make it more pathogenic? And for this, we weren't very successful when we tried to kind of overexpress individual virulence factors amongst those that we tried. But so we turned to look at transcription factors and found and focused on transcription factor NRG1, transcription repressor, which is actually heavily overexpressed in the commensal strain 101 compared to the virulent strain S2E3. Thus, we deleted NRG1 in strain 101, either deleting 1 copy and two copies, and we did this in close collaboration with Dominique Songlar from Lausanne. And found that actually then these mutants displayed an enhanced filamentation, already deletion of only 1 copy, so only a reduction of the NRG1 expression levels were sufficient for this, and this also correlated with an increased degree of agar invasion of this mutant 101 strain, as well as with the increased expression of a number of different virulence factors. And of course, we were most interested to understand what's happening in vivo in the oral mucosa, so infected mice, with the parental 101 and the deletion mutant. We could do the experiment only with the heterozygous mutant because the homozygous mutant tended to clump, and this led to a non-effective infection, meaning that fungal load was so much lower that we couldn't compare it to anything. So the fungal load was initially the same between the 2 strains that we wanted to compare, but interestingly and of note, this strain that was higher in filamentation and was actually, due to the reduced expression of NRG1, was actually able to induce an inflammatory response. Maybe this can be seen very nicely here in histology, was able to trigger some degree of inflammation. And this actually resulted in a enhanced fungal control and a reduced commensalism of the strain. To summarize, these data suggest that energy 1 plays a central role in the commensal lifestyle of Candida albicans in the oral mucosa, as it was also found to be the case in other tissues. Now, let me turn to the second part and ask a question about how is actually strain 101 controlled. So it definitely likes to be in this oral tissue, and it doesn't induce very much of a host response, which would clear it. However, is there any host response at all, or does the host see the fungus at all, or does it just not see it at all, given that really there is no sign of a host response early on? So again, we did transcriptomics, but this time we actually looked on the host side and found that while, as expected, and that's well known, the highly virulent strain induces a very rapid response, 101 actually initially doesn't induce any response, but eventually actually there is a very strong host response induced. This is just showing the overall number of differential expressed genes. And also, if we look more specifically at some genes of interest, and IL-17 is certainly one of them, we found a similar pattern, IL-17 being very strongly, very rapidly induced by the high virulent strain, but not by the 101 initially. But then eventually, 101 also induces a high degree of IL-17 expression. And from various numbers of different studies, we know, and also studies from Sarah Gaffney's lab, we know that this early IL-17 production is actually made by cells of the innate immune system, cells that are triggered in an antigen unspecific manner, simply by the triggering by cytokines, including IL-1, IL-6, IL-23. While later on, it's the adaptive immune response that kicks in. T cells that are primed in the lymph nodes that depend on antigen presentation, on co-stimulation, on cytokines for polarization, a much more lengthy process that is actually taking place in response to both strains, the virulent and the avirulent strain, to a very similar degree. So you couldn't tell apart which T cell response was triggered by what strains. And this is the T cell response in the draining lymph nodes, in the cervical lymph nodes on day 7 post-infection. So Candida-specific adaptive immunity is independent of the fungal virulence, it seems. And again, we showed this not just for these 2 strains, but actually many more high and low virulent strains. Now we were interested to look over time, because that's what matters really, the commensal phase in our view for the long-term stabilization of the equilibrium. And actually in response or during the commensal state in this 101 colonized animals, this T cell response stays very high up all the way, all the time during the time window we looked, and this is the case in the lymph node where the T cells are primed. But this is also the case in the tongue, actually the site where the fungus is present, where these 101 induced cells stay up very high, while in response to high virulent strains, they actually contract. And these cells, we analyze them by FACS. There are not so few in the tongue, as you can imagine. They express normal T cell markers, as we would expect. They express memory markers, and they make a whole bunch of IL-17. Now, let me just for a moment give you a little bit of background about T cells and what happens during time, or what we know is happening during time during normal infection, where T cell response, T cells are activated, expand, and then they differentiate in effector T cells, and depending on the infection, and we know that in case of fungi, they differentiate in Th17 cells. What happens as well is that these different subsets of T cells overall also develop into different types of memory cells over time of the infection. And there are different subsets of these, including circulating memory cells with cells that circulate only centrally through lymph nodes and blood, those that circulate a little further through blood and tissue and lymph nodes and back, and those that circulate, that don't circulate, actually, this other subset of resident memory cells, which actually stays in the tissue locally. And these are characterized by surface markers CD69, CD103. Now, we were interested to understand in our colonized tissues whether we find any memory cells and of what sort they are, and especially given the situation that is a bit different from the canonical way where you find T cells contracting upon resolution of an infection, while during commensalism, of course, there is no the trigger, the microbe remains, and also, as I've just shown you, the tongue T cells actually remain very high. So we looked at these tongue T cells in more detail and stained them for these markers that are characteristic for the TRMs, for these tissue resin memory cells, and actually find these cells to express very highly these markers, CD103 especially, and the IL-17 producers are really among these tissue resin memory cells, these CD103 high cells. So by phenotype, we found that the Candida-induced Th17 cells are tissue resin memory cells in the colonized tongue. However, there are more parameters that are needed to be fulfilled to really call them TRMs properly. What about their location? Are they really within the tongue tissue or just in the circulation that passes the tongue? How are they maintained, and whether is their function? So we looked first at the localization, and we used a little trick where you injected an antibody directed against T cells, especially CD4, into these animals shortly before we terminated. And in that way, the antibody only reached the cells in the circulation, but not those in the tissue. Afterwards, we isolated the T cells and then put another antibody on them, which could now reach all the T cells, those that were originally in the circulation and those that were originally in the tissue. And so we find in the blood, of course, all cells double stained for the antibody that we injected in vivo, as well as the one that we added ex vivo. But then in the tongue, those IL-17 producing cells were all singly stained, meaning that they have no access to the injected antibody and were really tissue inside the tissue. And then regarding the maintenance, we again injected an antibody against T cells against CD4, but actually one that depletes CD4 T cells that are circulating. And we waited a little longer and then assessed or for a control found that indeed all circulating T cells in the blood and the cervical lymph nodes were almost completely deleted, while actually in the tongue tissue, the CD4 T cells were still there at very high numbers, and the CD103 high ones actually even very much there. And in terms of their function, we found that these resident T cells that were remaining in these anti-CD4 treated animals were actually sufficient to control the fungus. And I think the significance of this graph only becomes clear if I show you in comparison the situation of T cell deficient mice, so mice that have absolutely no T cell whatsoever in any organ due to the absence of the RAG enzyme, or animals that have absolutely no IL-17 due to genetic deletion of the IL-17A and F gene, where we have 100 or over 100-fold of an increase in fungal load in absence of T cells and IL-17, while the fungus is nicely controlled if TRMs are still present. And the second approach was to not delete the circulating T cells, but actually block their access to the tissue. And this can be done with a drug called FTY-720, blocking the egress of the cells from the lymph nodes. And again, this without showing you the controls, but just the fungal load in the tongue. Again, this results in a completely well-controlled situation with no overgrowth. So altogether, we think these data illustrate that CD4 tissue resin memory cells provide local and are sufficient to provide local immunosurveillance of commensal Candida albicans. They express high levels of the typical markers. They produce Th17 effector cytokines. They're really located inside the tissue. They're independent of circulatory T cells, and they're sufficient to maintain the colonization. And these data are also supported, the data we find in mice, from a human study by Tom Cooper, who actually showed a few years back, when looking at human T cells, Candida-specific T cells in the skin of individuals, healthy individuals that are naturally colonized, where he analyzed the Candida-specific IL-17 producing cells and found that these also express TRM-specific markers. Now, questions that arise from this and that we're now looking at is the antigen specificity of these T cells, and especially the specificity over time. And there also an important aspect that we're wondering is whether these T cells are really memory cells the same way as memory cells that do contract and exist only in small populations. Lastly, whether actually the constant presence of such IL-17 producing memory cells have no immunopathological consequences if they're persistent really over so much time. And for the last, actually, we have, I can show you just one slide to give you a little insight. We have no evidence or no hint whatsoever that there will be any sign of inflammation or pathology over a course of up to a year. And also, if we delete regulatory mechanisms, immune regulatory mechanisms, such as regulatory T cells or the cytokine IL-10, this doesn't change and alter the Th17 population, nor does it really affect the fungal load, indicating that somehow these immunosurveying Th17 cells are really immunosurveying and have no consequence of immunopathology. Nevertheless, I'd like to close in the few remaining minutes by showing you an example where we have found a situation where commensal fungus-specific Th17 cells actually can have immunopathological consequences. And for this, I'd like to turn towards Malassezia and just give you very, very briefly remind you that Malassezia is really the most important and most abundant skin commensal yeast. Again, we established an infection model in mice. We use a model in mice where we can study the Malassezia host interaction, and very similar to the T cell response we find induced against Candida albicans, we also find the response against Malassezia to be a very selective IL-17 Th17 response in the draining lymph nodes, as well as in the skin, where we can also find these Malassezia-specific IL-17 producing T cells. And this is reminiscent of what's also found in humans by ourselves, but also by a very nice study by Geraldine and Alex Sheffield, where a large proportion of the Malassezia-specific T cells in healthy individuals are actually of the Th17 subset. Now, these Th17 cells, IL-17 production is important for fungal control, very much as in case of Candida. Certainly in our mouse model, I should note or mention that in contrast to our commensal Candida model, our Malassezia model is not such a stable model, and there the fungus does get clear in the current version of the model as we have it, but nevertheless, it allows us very much to say that the IL-17 pathway and axis is essential for controlling the fungus. If IL-17 is absent, fungus is overgrowing, and this is likely related to the regulation of antimicrobial peptides by IL-17. Now, in a condition where the barrier is disrupted, where there is some predisposition, some dysregulation of the homeostasis, we find that the immune response directed against Malassezia is actually provoking and exacerbating inflammation and causing immunopathology. And we demonstrated this by using or inducing barrier disruption in the skin of our animals prior to applying the fungus, and this resulted in a very strong exacerbation of the response, as you can see here by looking at the thickness of the ear. And also here a quantification, while the fungus alone or this mild tape stripping doesn't induce any sign of pathology measured here as ear thickness, the combination really induces a very strong inflammation, indicating that this Malassezia-directed host response really exacerbates pathology, and this is dependent on IL-17 and 23, so on this type 17 axis. Thus, this normally protective type 17 response can become pathological depending on the host conditions, the predisposing conditions. And given that our model is not a commensal model, we did a last experiment I want to show here, where we just applied a primary and then a secondary infection with the mice resting for a couple of weeks to let the memory response develop against Malassezia, and in mice which have been infected twice, so having induced a pre-existing anti-Malassezia response, this was actually then increasing the degree of the inflammatory response we found in tape-stripped and Malassezia-associated animals. Now, this is my last summary slide, just recapitulating that indeed in a homeostatic situation in a normal skin, the type 17 response against this second fungus on the skin, Malassezia, is relevant for control of the fungus and preventing overgrowth, while under altered conditions such as barrier disruption, this anti-Malassezia type 17 response it becomes pathological and exacerbates inflammation. With it, I'd like to close by, first of all, acknowledging my great team with present and past members, those working on Malassezia and those working on Candida and those working in between. I'd also like to thank dear collaborators, especially those who've been involved in the data I showed you today, Dominique Sanglard, Christophe, and many others, and of course also those who've contributed by providing reagents and mice, funding agencies, and most importantly, for your attention. I'm looking forward to questions. Thank you.
Thank you, Salome. So I'll turn it over to our co-chair, Dr. Geraldine Butler. And I think, Dr. Butler, you can probably take the chair's prerogative and ask your question.
Okay, I think you were taking questions for Salome, but maybe I'll ask my question anyway, which was, is overexpression of NRG1 a marker for commensal strains other than 101? Could you use it as a way of screening?
We're currently looking at that in more detail. I can't really say very much. It may not be very general, but I can't give a definite answer on that.
Okay, thanks. Okay,
I have a question for Betty, if I may.
Right.
Very nice talk, Betty. So one other receptor pathway, signaling pathway that's been implicated in netosis is the chemokine receptor pathway itself, such as CCR2 or CCR1. So do you think that pathway is also regulating nets in the peritoneal cavity in your model?
We didn't look at that, so I can't give you a straight answer.
Okay. All right. Thank you.
Okay, so maybe I'll start with questions for Betty, then we'll get back into order. And I'm very aware that it's past midnight for you now. So the first question is LC3 crucial for replication and survival of histoplasma in the macrophages?
We didn't specifically silence LC3 and study replication. So I can't really give you an answer. Yeah, we only know that it affects cytokine response. Okay, thanks.
So for Salome, we have a question about whether 101 also colonizes the gut, and does it establish commensalism in the GI tract in addition to the oral cavity?
Yes, it actually does. I mean, there is always a question, well, what is commensalism really? We clearly find it in the gut over all the time. We can detect it in the feces very easily. Whether it also really goes into the epithelium of the gut, this we haven't checked carefully enough that I could make a statement on that. For sure, it's in the lumen, and we also speculate that it's actually coprophagy that is necessary to actually maintain this long-term colonization.
Okay, thanks.
Excuse me, maybe Geraldine, I can give you an answer indirectly. We know that RS production, which is upstream of LC32 formation, RS does not affect histoplasma replication in a normal macrophages, unactivated normal macrophages. It's the iNOS mediated NOS production, which is activated by interferon gamma, that affects histoplasma replication. So if from answer your question from that angle, I would guess the LC32 formation does not affect histoplasma replication.
Okay, so that's an answer to the first question.
Okay.
Yeah. And we have a very long question here from Sean Zhang, which I'll try and ask, which is that FungiTel is a commercial assay which detects serum beta glucan. And you noticed that you said that C. albicans masks production of beta glucan, but they can detect BDG in patients infected with albicans. So does that mean they're detecting it from dead albican cells? Or do you think the Fungitel assay can also detect alpha glucan produced from live albican cells?
I don't know. Candida can shed alpha glucan. Well, in my test, we didn't look at alpha glucan on Candida albicans. We only know that beta glucan is not expressed in live Candida and is expressed after it is killed. So I don't know about alpha glucan presence in Candida albicans. Maybe someone else can help me with that. So with that, I can't really give a straight answer on the detection of glucan in Candida patients is alpha or beta glucan.
Okay, thanks.
Great. Salome, we have a question from Laura Chapello. So the TRM cells, have you looked at other strains, and are other strains that potentially could induce TRMs in an earlier time point, like as soon as 1 to 2 days? And also, I guess if you establish a gut colonization model first, does that alter the kinetics of the generation of TRM cells in the oral cavity?
Okay, I guess there are several questions in that. With other strains, yes, they're also very similar populations, and the way or the duration that they're maintained seems really to depend on the persistence of the fungus. So those fungi that get clear, every strain, every isolate has its own kinetic in the oral cavity, and the persistence of the T cells at these high levels depend on the colonization. We don't really see a more rapid induction. I'm not sure whether that question may relate also to the fact they're at a very early stage with the high virulent strains, also TCR beta cells that produce IL-17. So among these innate IL-17 producers, there's some which look like, alpha beta T cells. They look like Th17 cells. Sarah Geffen has found them. My lab has also found them to make IL-17. Sarah Geffen has called them natural Th17 cells. I guess it's still a bit unclear what they really are. They seem to, rapidly respond to cytokine stimulation. Are these really Candida-specific ones or not? I think we need to do more research on that to really know whether these are memory cells that get reactivated, maybe by cross-reactivity, etc.
Are they CD69, CD103 positive, like your later TRM cells?
We've looked at some markers and they have a different profile, but also it's of course not a black and white thing, right? It's not just on and off, it's also higher or lower expression. Yeah. So they're not exactly the same, but it might have to do that it's in the context of a much stronger inflammation at that time point, etc.
All right, thank you.
There was a part on gut colonization first. I'm not quite sure I got that part right.
Well, yeah, so the idea is, could Candida colonization at other sites affect the kinetics of the TRM pool in the oral mucosa?
Yeah,
so
we haven't done that. Yeah, we haven't done that.
Thanks.
Okay, for Betty, since CR3 doesn't activate NADPH oxidase and ROS production with histoplasma, do you think netosis in response to opsonized Candida, which requires CR3, happens in a ROS-independent manner?
So CR3 interaction with Candida albicans in what context?
I suppose, is netosis in response to Candida occurring in a by a ROS-independent mechanism?
Through CR3. You mean through CR3?
Through CR3, okay. Yeah.
Right, because we show that unopsonized Candida induced nets is through Dectin-2. So the question is whether CR3 downstream signaling pathway does not activate NADPH oxidase would induce netosis. Is that the question?
Yes,
Okay.
that's, yeah.
I don't think so, at least based on our study. It is NCF dependent. Of course, NCF deficiency reduces double-stranded DNA by half. It's not complete inhibition of netosis. So for that half, maybe is NCF1 independent.
Okay.
It's not complete abrogation of netosis in CR3 deficient cells.
Okay.
It's only
Thanks.
half of that's produced by wild type cells.
Great.
Okay.
Thanks, Ben.
Thanks.
Salome, we have a question from Peter Cook about the role. Do we know anything about the role of the innate immune system in maintenance of the TRM cells? IL-1, IL-23, IL-6, any of the, what's the interaction of the innate immune system with the?
We haven't done enough on that, Pete.
Yeah.
I'm sorry, you asked other questions before, which we haven't done yet. We're on it.
Yeah, I noticed that Jay Miskard recently had a paper, he studies pneumococcal TRMs, and he showed that actually epithelial cells, class 2 MHC epithelial cells appeared to be important in maintenance of the lung TRMs in his model. I wonder if the similar may occur, and who's presenting who has to express class 2 in your model too would be another interesting avenue of investigation, I think.
Yeah. I mean, what we know is that the fungus needs to be present, because as soon as the fungus leaves, and we can also do this with the 101 and clear it with antifungals, then this high level of the population goes down. Yeah. But yeah, lots of questions.
And so we have a question from Diego Rossi. I'm not sure I completely understand it. I think it's, could the response of macrophages be similar to dendritic cells?
In what ways?
So the question just says, could dendritic cell results be similar for macrophages? I'm not sure what specific response.
Like in terms of cytokine or the role of the relative roles of the C-type lectin receptors, is it the same in the dendritic cells versus macrophages?
Okay.
Innate cells may produce similar profiles of pro-inflammatory cytokines. There's overlap, and if dendritic cells stimulated by histoplasma, they can produce a profile of inflammatory cytokines as well as macrophages. But I believe that stimulation of different cells may, the same organism may use different receptors to respond, and there will be same outcome of some specific cytokine that you're looking at. But there may be not if you're looking at other cytokines. So it's a very, you need to do the experiment to find out what receptor is being utilized for what organism, for what function. So it's not a straightforward question for a straightforward answer.
Right. It's not simple.
Right.
Okay.
And then, Salome, we have a question from Johannes Westman about, again, comparing the 101 strain with the SC strain in terms of any evidence of cellular toxicity, LDH release, alarmin release, any of those things at early stage in the infection.
Yeah. So I've shown, well, some data from in vitro or experiments in culture where basically 101 doesn't do much at all, no damage whatsoever. EC1 expression levels are very low. There is filamentation also on the epithelial cells, but that's just not enough, obviously. And in tissue, we have, it doesn't look messy at all. It looks just like a normal, perfect tissue, and we have no sign of damage occurring.
And what's the consequence now that you have this substantial TRM population making IL-17? how does that impact the bacterial microbiome? It must have an impact, right?
Yes. Again, sorry, I wish I would be able to tell you. Obviously, the question is up there, and we're looking into it.
Definitely. Can I ask one more question of Salome? But on the Malassezia work. So, Malassezia overgrowth has been implicated in seborrheic dermatitis, right? But that pathology is really different than psoriasis, where anti-IL-17 drugs work. So, but I haven't really seen, in the psoriasis field, there's tons of RNA-seq data implicating, IL-17 in that pathway, but I haven't seen similar studies with seborrheic dermatitis. So, do we know anything about the molecular pathway that's activated in patients with seborrheic dermatitis?
No, not really. There is really comparably super little known on seborrheic dermatitis, and this cause-consequence relationship also of Malassezia, the anti-Malassezia response, and disease pathogenesis is really quite unclear.
Yeah.
But we hope to use the mouse model to experimentally be able to maybe, at least in this model, give some hints. But yeah, from the clinical standpoint, also, compared to Candida, obviously, IL-17 doesn't seem to be that essential for control as it is for the control of Candida albicans. But it might also be more difficult to see, as there is not just fungal biofilm occurring.
Eddie, just a technical question for your talk. What's your opsonization procedure for Candida?
It's just incubate the fungus in a 10% serum containing medium.
Okay. All right, we're getting toward the end here, but we have two late questions, which I'll ask for Salome. Well, one from Joe Heitman. Are there any patient populations being treated with newer specific immunomodulators that have increased risk of Malassezia-associated skin disease or systemic infection that you're aware of?
Not big studies. I think there was something about IL-23, but I would need to look it up before I say anything that may not be correct.
And the other question was related to TRM responses to Malassezia in germ-free mice. So is there, what would you get? Yeah, go ahead.
We haven't looked in much detail also because our Malassezia model is not such a stable model yet. We still hope to manage to get it more stable or more colonizing. So we haven't properly looked yet, and we haven't really looked in germ-free mice. Antibiotic treatment doesn't seem to affect the Malassezia colonization to the level that we've looked at.
Great. All right. Geraldine, should we close it?
Yes, I think we've got everything.
Yeah. Thanks for Betty and Salome for two excellent talks, and we hope to see you all in November for the November MycoTalks.
Thank you.
Okay.
Thank you very much.
Okay, bye.
Okay, bye.